Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Advancing Imaging Operations and Analysis in Alzheimer's Disease Late Stage Clinical Trials. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank IXICO who developed the content for this presentation. IXICO is a leading neuroimaging provider whose purpose is to advance medicine and human health by transforming data into clinical meaningful insights. IXICO provides advanced brain imaging and AI-driven analysis solutions to enable better trial design, patient selection, and outcomes across all phases of clinical research. Their neuroimaging services are deployed in some of the most important clinical trials in neuroscience, lending insights into disease progression and helping sponsors maximize the value of their drug development programs. Now, it is my pleasure to introduce you to your speakers for today's event. And first, I'd like to introduce you to Roger Bullock, Chief Medical Officer at Oryzon. He has extensive experience as a clinical researcher, having participated in more than 70 trials in Alzheimer's disease and other conditions. Dr. Bullock is considered a world KOL in the space of neurodegenerative diseases. Over his 30-year research career, he has authored and co-authored more than 100 peer-reviewed publications and book chapters in this domain and presented at numerous conferences. Next speaker is Shona Clegg, and she's the Associate Director of Client Services at IXICO. And Shona joined IXICO in 2016 and has managed clinical trial projects for clients in a variety of phases and therapeutic areas, including Alzheimer, progressive supranuclear palsy, and Parkinson's disease, across a wide range of protocols encompassing MRI, DMRI, DTI, functional MRI, and PET imaging. With her extensive operational experience of on-time and on-budget project delivery, Shauna's current role includes quality and service management to help clients achieve their imaging objectives with the utmost quality performance. And next is Richard Joules, and he's the Imaging Science Manager with IXICO. And Richard has worked in a range of therapeutic areas such as Alzheimer's disease, Huntington's disease, progressive supranuclear palsy to develop methods for biomarker detection and disease monitoring. As the current Imaging Science Manager, he is responsible for deployment and monitoring of analysis solutions in clinical trials. Now, before I pass the mic over to our first speaker, and that's Roger, we have two poll questions for our audience members to interact with in real time. So that's appearing on your screen momentarily, and there it is. So as I said before, this is done in real time, so your participation is greatly appreciated and encouraged. And the question that we have for you today is, what is your biggest pain point when it comes to Phase III Alzheimer's disease trials? So please go ahead and select one of the below. And your options are choosing correct imaging endpoints. The other option is defining patient population or heterogeneity of enrollment subjects. And that's heterogeneity of enrollment subjects, and managing growing complexities of Phase III trial. So please go ahead and select one of the below. And it looks like we have the majority of our audience members have done so. So thank you very much for that. If I could please get the remainder to go ahead and cast your vote, I'll give you 10 more seconds and then I will close the polls and we'll get to the second poll. Okay, thank you everyone for that. I'm now going to close the first poll and share the results with you. Okay, so here's the results of our first poll question. And the question again was, what is your biggest pain point when it comes to Phase III Alzheimer disease trials? And we have 29% saying choosing correct imaging endpoints. Then we have 5% at defining patient population. Then we have 38% at heterogeneity of enrollment subjects. And then we have 29% at managing growing complexities of Phase III trial. So thank you everyone for participating in that last poll question. And now we're going to launch our second poll question, and that should be appearing on your screen momentarily. And there it is in front of you. And just like before, we're going to do this in real time. I'll be very quickly about this one here too. I'll give about 40 seconds for this poll question. And the question is, what do you see as the main challenge when defining the PET component of your clinical protocol? So please choose one of the below. And your options are choose a choice of PET tracer manufacturer, choice of visual read versus quantitative analysis, or phantom requirements for standard qualification, or the last option, choice of PET rating scale. So please go ahead and cast your vote. Okay, thank you, everyone. Looks like we've done that. Great. Thank you so much, everyone, for participating in that. I'm now going to close that poll and share the results with you. Okay, so here are the results of the last poll. And that question was, what do you see as the main challenge when defining the PET component of your clinical protocol? We have 6% at choice of PET tracer manufacturing, 78% at choice of visual read versus quantitative analysis, 11% at phantom requirements for scanner qualification, and 6% for choice of PET rating scale. So thank you everyone for participating in that last poll question. And now I'm going to pass over the mic to our first speaker, Roger. So Roger, when you're ready, you may begin.
Thank you, Sonia, and welcome everybody. Pleasure to be taking part in this symposium. Next slide, please. So I'm just going to give a brief overview because the meat of the talks is the following too. But I'm going to just talk about the current position of what we've got on offer and then some of the key challenges, which is interesting because I think that poll has justified some of the points I was going to make. So next slide. And again. So I'm going to start just in putting into context, the ATN classification, which we've come to use in clinical trials, in Alzheimer's trials. We're pretty good at the A and we're pretty good at the T, and I'll mention the PET scans in a minute. But N is not really clearly defined, and neurodegeneration we can see structurally on MRI, and we can measure in various ways. So maybe we need to spend a little more time thinking about the N. For example, with MRI, I mean, can you get a better quality of heterogeneity with using an MRI biomarker as well as the other biomarkers? And maybe even do it first. Maybe, if we can get really early signs of neurodegeneration, then we might actually get better patients to enter into clinical trials and even predict those that might decline quicker so that we can get a better placebo effect. What we've got at the moment is amyloid PET and tau PET, which are both now in the clinic. We've got SV2A PET, which is looking at synaptic loss, which I think is very interesting. It's a bit early yet, but given the very early papers on Alzheimer's showed that most of the disease correlated to synaptic loss, then it will be nice to be able to measure that in more detail. And then we've got MRI, which I think to a certain extent MRI, because of new things have come along, has been left behind a little bit, but certainly structural and functional MRI, they're reasonably well understood. But we've also got, free water imaging, which is particularly good in hippocampal atrophy, which, given that's one of the early structural manifestations of Alzheimer's disease, it's worth exploring a bit more. And then MRS, where we can actually get down to biochemical information. And, if we put all that together, then we ought to be able to classify patients maybe at baseline differently. Next slide. So amyloid PET, amyloid PET's been around a while now. I was back in the day with PIB, and of course one of the problems with PIB was you had to make it on site, and you had to be ready within 10 minutes, and a lot of patients travelled 20 or 30 miles and didn't get a scan because of the problems with the tracer. It's much better now. And we know that the amyloid PETs more or less correspond to pathological distribution. They're now used diagnostically in terms of for clinical trials and generally accepted. But, amyloid doesn't correlate well with cognitive decline. And as we've seen in recent studies, way back as far as bapineuzumab, removal of amyloid in regions of interest does not always coincide with any particularly clinically significant change. So, it's good to do, it's well accepted, and it helps produce a stable population, but it's not a cheap test, and it hasn't yet produced clinically meaningful results. Next. And now we've got tau, which we do know follows Braak staging, and it does discriminate Alzheimer's from non-Alzheimer's. And interestingly, in several studies, it's outperforming the CSF measurements. So that's good, and it also correlates to cognitive loss. However, it's very early in its development. There's still some off-target binding, and at the moment there's an unclear relationship with other biomarkers. So now we're going to have to see what happens if you have amyloid and tau scans, and then see how we work with that. We suspect one follows the other, and we may get more information over time with a bit more of temporal relationship. Next. So, as I said earlier, SV2A, that's very early in its development, and it needs more replication. But, it's a useful tool for measuring synaptic loss in the hippocampus, along with MRI, which is also very good at quantitative analysis of hippocampal volume. That may be a better way of screening people rather than just amyloid or tau positivity. Next. So MRI, structural MRI, we're going to be hearing a lot more about MRI in a minute, but it's been around a long time. We know the brain atrophy patterns. The hippocampal volume is a very early measure and very stable measure of the normal through MCI through Alzheimer's disease. Volumetric MRI, long been used. And you can also now start doing things like with shape analysis and measure ion deposition. Next slide. And then functional MRI, you can start looking at connectivity and more distinct patterns of atrophy. And I think it's generally accepted now that arterial spin labeling is probably as much, gives you the same information as FDG PET. So that at least reduces the need for that. And then we can look at white matter and also do multiple tests at once in the scanner. And then there's to analyze the data, you can use graph theory, which connects both function and structure, and machine learning models. So there's a lot of information you can get from MRI. Next. Okay, and again. So that was a next slide. That was a good rundown of, well, very quick rundown of most of what's available to us at the moment. How's it going to help? Well, clinical studies are. Can you go back a slide, please? I think I probably said next slide twice. Yes, sir. So we've got the tools available to us, but we've still got multiple challenges in terms of setting up clinical trials. And part interesting development out of COVID has been, which sites and which, I mean, I'm not going to talk about virtual trials or anything at the moment, but, the sites in hospitals tended to lose more patients, in public hospitals tended to lose more patients than those in private sites, in our experience, because they weren't going to a hospital site. A lot of the imaging was within private sites, and the retention in those sites was better than in the public sites. Obviously obviously going forward, we're going to have to be pandemic proof. Even if there is a vaccine fairly shortly, the trials that are ongoing and trials that are planned next year are still going to have to be shored up in many ways. Another problem with site selection is KOLs. It's vast unspoken, but a lot of KOLs, they are the PI in name, but they don't do a lot of the work. And also, there's some problems always with adherence to protocol. That's not so much around imaging, but if you've got 5 studies and 5 different ways of doing one of the cognitive tests, errors do occur. So there are problems around site selection. Patient recruitment is always a thorny issue. We've got many companies making a lot of money out of patient recruitment, but I think a lot of it depends on selection strategy. We've got various initiatives trying to get pre-worked up patients. I don't think the recruitment from those pre-worked up patients is as good as people hoped it would be. And then we've got issues around stratification. So as we learn more about Alzheimer's disease and some of the different things within it, we stratify for E4, but I think we'll be stratifying for much more, because it's fairly clear that this is definitely that Alzheimer's disease is a collection of diseases. If we start stratifying too much, then the sample size goes up. So, this issue of heterogeneity is key, and it may well be that some treatments work very well for some aspects of Alzheimer's disease, and by not segmenting at the start, we may be missing important information by doing clinical trials on a very heterogeneous group. Can we do better? Well, everything's done cross-sectionally. I know we're trying to get banks of patients and follow them up, but I suspect that we could do better. We could do some longitudinal running. Maybe just cognitive and not so much of all the other workup. And the other thing is modifiable risk factors, because it's now fairly clear that certainly exercise and diet have an effect on cognition. The Souvenet 3-year data has continued to improve on a 2-year and a 1-year. So, and the people are getting more clued up now. So older people, they know to eat better if they can, and to go for a walk once a day. And if you've got 1,000 patients all doing that to a different effect, then even without the heterogeneity of disease and pathology, you might have a heterogeneity of people adjusting their modifiable risk factors. So can we do anything with that? I mean, maybe technology, smartphones, maybe we can get people to maximize their baseline potential so that they are a bit more equal. And then when you get to the patient and caregiver themselves, then there's expectations and managing to see through 2 or 3 years of extensive visits and interviews. I think expectations is crucial. In the very early days when I started, you'd have people queuing up for Alzheimer's trials. They were much simpler. And their main motivation was they wanted to do something to help their children and families, rather than something for themselves. Whereas now, if we're right about some of the experiments that we're doing, then actually, if you're in your 70s and it works, there's a very real chance there could be an impact on your own disease. So we have to be very careful about the expectations and what we tell people. And the one elephant in the room always is, and always will be, is that Alzheimer's disease is synonymous with dementia. And actually, a preclinical Alzheimer's disease, or even a prodromal Alzheimer's disease person, are functioning very well. And I think we lose some people because they don't want to hear the words Alzheimer's disease, and therefore they don't sign up for these very early experiments. So, and also, I suspect that investigators give a different explanation to the patients. So, some are over positive about what they're trying to do. I mean, you can't be negative, but And if you set the expectations too high, and then 6 months into a trial they're not being reached, then I think it causes problems. And I have discussed with people about scripted interviews for consent, but it's a minefield, especially in the US. Next slide. And then when you start looking at the main practical issues, the number of assessments. I mean, studies just get bigger. Very little gets taken away at the moment. And I don't know quite how we tackle that, but I think we need to have some proper discussions around it. I mean, some of the tests that haven't worked in any studies, maybe they're not the gold standard, and maybe we should be looking at new ideas. Now we're getting to the stage of the number of scans. I mean, we've got studies now with amyloid and PET scans, and what happens when we get new PET ligands? I can't imagine that if we get to 7 PET ligands, we're going to do them all in one study. So I think as we get more knowledge and start selecting tests around imaging, we really need to be thinking about what we think the drug's doing, what we think where the main results will be, and that there'll be challenges for the future. As I said at the beginning, big fan of MRI. I think there are a lot of utility. You can do a lot of things at the same time with it. The analyses that are coming out now are much more than just a picture of the brain. You can look at over time. There are algorithms for predicting decline. And I think MRI is due a bit more of a comeback and a bit more emphasis in the screening and eligibility, not just the safety. The other thing with clinical trials now, of course, is the cost. And every company in the world is looking at how to minimize costs while preserving power and accuracy and protecting the endpoints. And we still don't know what the best endpoints are. A lot of our endpoints, certainly in Alzheimer's studies, are heavily reliant on the caregiver. The biomarkers are not as precise as they might be in terms of relation to clinical improvement. So what can we do? I mean, can we get into real-time assessment rather than visits every 3 to 6 weeks? Can we can we start to leverage smartphones and other equipment to get a continuous monitoring? We all know that patients have good and bad days when they come to clinic, so maybe there's better ways of doing it. And as I said several times before, we need to think about predicting decline. So last slide. So for neuroimaging, I think what we need is a good balance between innovation and service. We need to be thinking how to do things differently. Developing novel models for eligibility, safety, and outcomes, rather than just safety and outcomes. And this will be relevant for both research and future clinical practice. The safety packages of the biological compounds is going to include ARIA and various things. So we need to be thinking about scaling up around both measurements. So we do need to evolve scalable solutions for what's going to be now the next era of AD therapeutics. And that was a very quick run through of some of the challenges and what we've got. And I'll stop there and hand back to Sonia. Thank you.
Okay, thank you very much, Roger. And we are now going to launch our third poll question. So audience members, you'll see it in front of your screen, and there it is. And this poll question is, what would be of most benefit for imaging sites during study setup? So we have a couple options below. We have one being partially pre-populated site qualifications, survey forms, online training, resources for site, summary overview sheets for technical staff, or technical help desk for site queries. So please go ahead and cast your vote, and then I will share the results with everyone. Okay, it looks like we have the majority of our audience members have cast their votes, so thank you very much for that. And I'll leave another few seconds for the remainder to go ahead and cast your vote right now. And okay, perfect. Thank you very much, everyone, for participating in that last poll question. So let's take a look at the results. Okay, here are the results. So the question was, what would be of most benefit for imaging sites during study setup. And we have 30% at partially pre-populated site qualification survey forms, 40% at online training resources for site, 25% at summary overview sheets for technical staff, and then 5% for a technical help desk for site queries. So thank you everyone for participating in that last poll question. And now we're going to pass over the controls to our second speaker, and that is Shona. So Shona, when you're ready, you may begin.
Thank you, Sonia. Thank you, Roger, for your presentation and everyone for joining. So for my section of the presentation today, I'm going to be talking about imaging site qualification optimization, data management, and radiological reads. So the first section is imaging site qualification optimization, and this section covers both MRI and PET. So there are quite a lot of considerations that can be helpful and beneficial to the protocol design and the setup of imaging within your clinical trials. The first section being MRI protocol development. So it's important to consider right at the protocol development stage exactly what the analysis goals are for the study, and this allows for support of these goals in the parameter selection. What we want to do is to be sufficiently specific to allow us to have each site be able to meet the requirements for the study. It helps if we can be flexible enough to accommodate a variety of different scanner platforms. So that would be different scanner makes, models, coil strengths, software versions. We generally find that what works well across studies is to use the three primary scanner platforms, so GE, Philips, and Siemens. Another consideration is the field strength. Are we going to focus on 3 Tesla, or are we going to allow a mix of 1.5T and 3T scanners into the study? And also deciding on the number of head coils that we think will work best within a study is an important thing to get agreed at the start, because it helps with the selection of sites. Some of the challenges and complexities we come across can be exemplified through the 3D T1 weighted images. So there are a lot of parameters within these sequences that can have significant effects on the scans themselves. So for instance, contrast parameters of inversion time, echo time, and repetition time all have a significant effect on the contrast of the scans, and which may then have a knock-on effect on the analysis that is being done. We also need to consider the resolution of the scans themselves. What voxel dimensions do we want? Do we want isotropic voxels? And how does this impact on either the reads that are being conducted on the study or the analysis goals for the study? Looking at the acquisition and reconstruction matrix is also important, and trying to decide whether we want to do things like acceleration on acquisition to potentially speed up these scanning times. Another thing that's important to consider is whether we want to try and harmonize these scanner-specific parameters across the manufacturers' makes and models to allow us to get the most even balance, and to allow the analysis to be as comparable across those platforms as possible. So all of these things go to show that knowing the analysis goals at the outset allows the decisions on these parameter selections to go a lot more smoothly. In terms of image resolution, we have to consider the trade-offs. So does the benefit to the analysis, does the analysis benefit from increased resolution or not? If there's no benefit to the analysis, then we should consider whether or not we want to spend the increased time that more detailed resolution requires from scanning. Is it feasible to harmonize the different scanning resolutions across all scanners, especially key if we've got a mix of 1.5 and 3T scanners within a study? And as I kind of alluded to just now, the increased scanning time or decreased signal to noise ratio, we need to decide whether that is a benefit for the analysis we're doing in terms of the increased times for the scanning. Once we've set the protocols and harmonized the sequences across the different scanner platforms, we then actually move on to the scanning itself. And there are quite a lot of issues that can impact the scans. So what we find are some of the most common issues that impact the longitudinal comparability are changes to the scanner. So this is either software or hardware upgrades on the scanner, or changes to the qualified sequences where sites have made a tweak or added additional slices to the imaging to allow for a particularly large head, for instance. All of these can have knock-on impacts on the analysis outcomes and also potentially on the radiology reads as well. Aside from that, there are scan and subject-specific quality issues. So the most common of those across most disease groups would be motion artifacts. There's also non-linear scaling, where we see warp of longitudinal data with subjects positioned differently within a scanner. Noise within certain areas of the image can be a problem, and also wrapping, where the nose can be wrapped into the back of the image, causing degradation to some of the slices of the brain. Another issue that occurs is missing slices. So how can we mitigate for these issues? Initially, we recommend that you gather scanner and site details. So this will allow decisions to be made and sequences to be harmonized across the scanners that are actually going to be running within this study. It also allows scanner-specific protocols to be provided. And then we'd always recommend ensuring the correct setup of sequences through a phantom or healthy volunteer scan as the protocol dictates, just to make sure that everything is set up correctly before the first subject starts some scanning. Patient positioning is another key area. The proper positioning is really important for the successful reproduction of serial MRI scans. Ideally, the subject has suitable padding around their head to minimize movement. The field of view planning is, again, another area where we find a significant focus in our training has to come. It's really important for ensuring that we avoid the non-linear scaling issues in the longitudinal scanning. The longitudinal consistency is critical to ensure that we have accurate measures of change. And as part of this, in terms of quality review, we'd always recommend and include in our training for sites that they review the images at the time of acquisition to allow them to rescan any sequences that aren't up to quality while the subject's still in the scanner, therefore reducing patient burden by having to be called back for additional rescans. Once that data is then available to the central imaging vendor, we would always recommend automated assessments of scanning parameters. These are extensive lists of quite detailed values and not the kind of thing that you can eyeball. So an automated assessment of these is always far preferable. Also, tying in with the field of view planning, we recommend automated assessment of positioning within the scanner, and then a visual assessment of the quality of the image to detect the motion artifacts I described earlier, or more generic artifacts like signal coil dropout, and the swift review of these and feedback to the sites allows any modifications that need to be made by the site to be made in real time. And then the final section of mitigation is monitoring. And this is basically to actively keep contact with the sites so that any hardware or software changes that are upcoming, the sites can let us know and can engage with us early, so we can start to work with them on what kind of impacts they might have. We can also suggests in some cases, depending on the analysis types, it's worth looking at getting healthy volunteer assessments before and after an upgrade to allow quantification of that change. Moving on to data management. We find that having a centralized digital platform for all of the uploads and tasks works really well. It facilitates swift review of data, transfer between sites, readers, and sponsor, and allows us to streamline the processes. So what we find works very well is for MR images to be uploaded and PET images from the site into the software. Then image quality control and metadata parameter checks can be run through that and triggered automatically. When suitable images are passed, they can then automatically move into the read and or analysis pipelines. And when the radiology reads are completed, reports and notifications can be sent out directly through automated email notifications. And they can go either directly to the site, the sponsor, or a mix of both. So focusing in a little bit more on part of that quite complicated diagram from the last slide, we're going to track a scan through the process. So the scan is uploaded by a site and it receives a quality control review. This is several stages. So there's the automated metadata QC, checking the parameters of the scan, making sure it was done on the correct qualified scanner. A longitudinal stability QC, so this is checking the positioning of this scan and any earlier scans within the scanner to make sure that we're going to avoid gradient warp or issues like that. And a visual quality control step, looking for gross errors such as motion artifact. When the image passes all of these checks, it would automatically move into the next stage of the process. Richard will be taking you through some of the analysis suggestions, but here we'll focus on the radiological review. Once the images have passed through the quality control steps, they move into the radiology pipeline, where images and read forms are sent to the reader, who can then view those directly and complete their radiology reads. This data is then all stored within the system at all times, and so the system can then automatically send out the reports to the site, send the eligibility reports or the safety reports in real time, and this allows for expedited eligibility checking and enrolment of suitable subjects into the studies. Another part of the data management, and I think this ties in quite nicely to the poll results, is allowing all the key documentation to be stored within one place. So what we find works really well is having all the site documentations available through the web-based interface. So all those site users with access just need to log in to this one location to access the site operations guide, the upload instructions, the scanning parameters, and then they have access to that all in one place rather than needing to delve around and locate the study file at the site. Especially useful as imaging sites are often not part of the clinical site directly. There's an image upload section which allows the image technologists or data managers to conduct the uploads, but also allows the study coordinators to see which scans have been uploaded and which are pending or overdue. And we find that that facilitates the links between the clinical and the imaging sites in terms of data. We also have a data queries section, and that allows the queries on imaging quality, the requests for rescans or re-uploads to be viewed by the site staff. It again facilitates that information flow within the site, which may be split between clinical and imaging sections. Moving on to a little more detail on the radiological read process. So as I've mentioned earlier, The images undergo quality control, and then they're pushed to a dashboard, which is where the central readers can see all of the reads that are pending their review. This allows them a very clean space to access these images without having to navigate deep into complex databases or systems. They just need to click on one row within a dashboard. It also allows for the prioritization of scans. So if we have reads that are particularly urgent, they can be flagged up for the readers. When the read is selected, there's an integrated image viewer which can be used to view the images from this visit, and if it's a longitudinal follow-up visit, all of the earlier visits for that subject. And the same for the radiology read form. They will see not only the current read form, but the historical read forms as well, which works very well for facilitating the review of historical data and tracking the progression or resolution of change over time. The read form then has a QC step just to make sure that everything's filled in, and then that gets automatically emailed out to the sites, the sponsor, the data management board, or whoever's been configured to receive those. This allows for an efficient single central read process, providing a rapid turnaround time. As I said, the access to historical data allows the readers to follow the changes or update new findings and identify which are new and which are existing findings that have previously been identified. The automated reporting allows for the rapid decision making on eligibility, getting those read reports back to the sites as soon as possible. Excuse me. It also allows us a good system to support the resolution of any local read discordance and a decision-making system with that. And moving on to my last slide, looking at PET read forms and our recommendations on standardizing these. So we have an example read form on the right and an example of the Tau rating scales that we recommend. So these are the presence on the, the presence of uptake on the severity scale of 0 to 3 in cortical regions, in BRAC stage composites, and also the presence of uptake in neocortex regions to help us identify scans inconsistent with a diagnosis of Alzheimer's disease. Also, the benefits of having a centralized PET read process is it allows us access to a pool of expert readers who have been trained in a standard read approach with inter and intra-rater variability against a gold standard. As with the MRI reads, it allows for a fast turnaround and direct reading within controlled GCP compliant platform. It also allows us to provide the images and consultancy for data management, data safety monitoring board, and to tweak forms as required for studies. I will now hand back to Sonia.
Well, thank you very much, Shona, for your part of the webinar presentation. Now we're going to do our last poll question. I'm going to launch it right now, and there it is in front of you. What is the most important when choosing image analysis pipelines? So we have your fully automated algorithms, most up-to-date advanced algorithm available, off-the-shelf widely used software, or therapeutic area specific validated algorithms. Okay, so please go ahead and cast your vote. I'll give you a few more seconds. Okay, perfect. Thank you very much, everyone, for participating in the last poll question. Now I'm going to close the poll and share the results. Okay, so here's the results of our last poll question. What is most important when choosing image analysis pipelines? And we have 6% with fully automated algorithms, 29% with most up-to-date advanced algorithm available, 0% for off-the-shelf widely used software, and 65% for therapeutic area specific validated algorithms. So thank you very much, everyone, for participating in that last poll question. Now we're going to pass over the controls to our next speaker, and that is Richard. So Richard, when you're ready, you may begin.
Hi, thank you, Sonia, and thank you, Shona. For the next few minutes, I'd like to focus on volumetric analysis of structural MR images, specifically looking at some of the core biomarker regions typically employed in clinical trials, so primarily the hippocampus, but also the whole brain and lateral ventricles. Volume measures and volume change measures in these regions have been assessed with several established methods within the literature and in previous trials, such as the BSI, Sienna, and 3-Cepher pipelines. These methods have demonstrated good sensitivity and accuracy and reproducibility, but may not necessarily have the flexibility to adapt well to new regions of interest, and can sometimes require manual intervention in order to achieve optimal results. And this may take the form of semi-automated initialization, parameter tuning, or post-processing and segmentation refinements. At IXICO, we employ fully automatic multi-atlas based approach called LEAP, which we've successfully deployed in several clinical trials over different therapeutic areas. In this pipeline, in brief, T1 images are pre-processed, skull stripped, and in homogeneity corrected. Input images are then compared to a reference database of T1 images with pre-existing segmentations for the region of interest or the parcellation schema. These segmentations for the most similar database images to the input selected in a manifold space are transformed to the input image, a majority voted to create a consensus initial segmentation, which is then automatically refined. This provides robust, fully automatic cross-sectional segmentations, either of individual regions or whole brain parcellations without the need for manual intervention. Volume changes can be estimated with a subsequent follow-up pipeline, which employs 4D graph cuts. And in this approach, regions of interest are simultaneously segmented in a follow-up and baseline image, with segmentations being temporally coupled and initialized by the baseline cross-sectional segmentation. And this provides us a way of measuring volume difference between these simultaneous segmentations, which we can then use as a volumetric endpoint. So this approach has the advantage of being both fully automatic, so there's no manual intervention involved, but also very flexible, allowing us to update the reference database that generates the consensus segmentation to include subjects from different therapeutic areas or different populations to best fit the study under investigation. We can also easily update the parcellation schema to include new regions of interest that are disease relevant. And this is what you see here, an example of the neuromorphometrics whole brain parcellation schema being updated for progressive supranuclear palsy in the brainstem. This approach can also be modified to segment individual regions of interest for highly optimized volumetric estimation. And this is exactly the approach we've applied to the core AD regions of interest, so the hippocampus, whole brain, and lateral ventricles. Starting with the hippocampus, we validated a implementation of LEAP against a gold standard consensus-based hippocampal dataset. And in this protocol, 2 expert raters manually segmented the hippocampus for 135 subjects. And these were merged to create a gold standard reference dataset, which we used to validate the pipeline. We see very good spatial overlaps, as shown by the dice scores in the lower left, as well as good agreements in absolute volumes and significant correlations. Notably also, the variability between the manual and the automatic segmentations is in line with inter-rater variability. The variability, in addition to direct comparison of volumes, we also validated LEAP in terms of accuracy in predicting 2-year conversion from MCI to AD diagnosis using empirically derived threshold. Also considered were the MTA scores and lateral ventricle volumes as predictors for conversions. And hippocampal volume was found to be one of the most sensitive predictors to conversions with the LEAP approach providing comparable accuracy for prediction of conversion within 2 years to manual segmentations. And details of that can be found in the paper reference there. In terms of volume change, which is a key biomarker in many trials, we validated the LEAP pipeline against manually initialized or semi-automatically initialized BSI measures and free surfer for a 2-year decline in 100 subjects. We compared the pipelines in terms of sample sizes needed to detect a 25% change in atrophy values for a hypothetical 2-arm clinical trial. And we see that BSI and LEAP perform significantly better than the free surfer pipeline, as shown in the table, specifically the last column showing sample sizes, with LEAP performing only marginally worse than the BSI. However, it's worth noting that LEAP is fully automatic and flexible, whereas the BSI was manually initialized and potentially susceptible to vulnerabilities of intra and intra subject variability for those initialization segmentations. Also being fully automatic, it has much reduced resource cost for deployment, meaning much faster turnaround times in terms of volumes. For the whole brain, we did a similar validation comparing the LEAP implementation against semi-automatically initialized BSI, again showing comparable results with the BSI with significantly correlated volume estimates and sample sizes, again necessary to detect 25% change in atrophy, as shown on the right, as well as sensitivity to predict clinical groups from volume alone, as shown in the lower table. And very quickly as well, lateral ventricles were also validated with a very similar approach. Again, semi-automatically initialized BSI was used as a reference method, and we see that LEAP performs comparably in estimating absolute volume changes, but also in terms of group separation, where there was no significant difference between the methods. So in these slides, we've demonstrated that the LEAP method performs comparably against a commonly employed standard, so the BSI using best practice semi-automatic initialization. However, LEAP has the advantage of being fully automatic, flexible, and scalable to allow therapeutic specific validation in clinical trials. In the next few slides, now that we've seen the tools, I'd like to discuss some of the applications of these, specifically with the examples of using hippocampal volume in efficacy analysis and patient selection or stratification and sub-analysis. So in order to assess treatment efficacy, historically clinical rater scales have been used as an endpoint. So whilst cheap and simple to administer, they are very coarsely granular and potentially insensitive to subtle effects. However, quantitative volumetrics provide a much finer granularity, which shows significantly greater sensitivity to efficacy, as demonstrated here in a table comparing ADAS-Cog, MMSE, and hippocampal volume and whole brain necessary sample sizes necessary to detect, again, a 25% change in a 2-year clinical trial, and details can be found in the paper linked in the bottom corner. Really demonstrating the use of quantitative measures for reducing sample sizes, and this has obvious downstream implications for patient recruitment feasibility and clinical trial costs. Also, as mentioned in a previous slide, hippocampal volume has been shown to be a sensitive predictor of cognitive decline. Specifically, the normalized cross-sectional hippocampal volume can be used as a feature to assess whether a subject will likely convert from from an MCI to an AD diagnosis within a 2-year period, using empirically defined thresholds from independent data. In this example on the right, thresholds were estimated from an ADLI data set. This finding is robust across multiple methodologies, but with LEAP being one of the leading 4 methods assessed in the paper on the right. So this indicates that hippocampal volume is a good candidate for enrichment, using it to identify patients with likely short-term decline, for inclusion in clinical trials and getting potentially much greater power for efficacy and placebo analysis. So here on this slide, we show the impact of employing such enrichment in a clinical trial setting. We compare the effect of enriching with hippocampal volume to using amyloid positivity as a screening criteria. The graphs on the lower half of the slide show for an MMSE and ADAS-Cog outcome measure in a hypothetical 2-year clinical trial. The sample size is needed to measure a 25% effect on the right, and on the left we see the SNR, which is the signal to noise, which is a measure of the 2-year change in the outcome measure divided by the standard deviation, and it informs the power of the signal of our interest. And overlaid on both plots, you see a line in blue, which is the expected screen failure rate. So moving along the bars from left to right, we have our reference unenriched population with associated SNR and sample sizes. By enriching with low hippocampal volume, so subjects with a normalized hippocampal volume less than a data-driven threshold, which put them at risk of likely short-term neurodegenerative decline, we see improved SNR and sample sizes, as shown by this N+ bar group. Another commonly employed enrichment criteria in more recent trials is to screen or perform stratified analysis based on amyloid positivity, and this has an even greater effect on improving SNR and reducing sample sizes for clinical trials. What's important to note, however, is that this neurodegenerative enriched population is not necessarily the same as this amyloid enriched population, which allows us to combine both screening criteria to create a amyloid positive group with likely short-term neurodegeneration. And this can result in a greater than 50% improvement in the signal to noise ratio and sample sizes compared to an unenriched population. So you may have noted that this also results in a expected screen failure rate of around 45%. However, due to the increased power of this enriched population, you wouldn't necessarily need to screen more subjects to power your clinical trial, as shown on the left. And in fact, you would enroll fewer patients in a longitudinal trial, and therefore, over the course of 2 years, your trial costs would be significantly reduced compared to screening on amyloid positivity alone. And again, further details can be found in the papers cited in the corner. So finally, my last 2 slides. No talk regarding MR volumetrics would be complete without at least mentioning AI, or more specifically, deep learning. There's been a strong recurring trend in the field to employ deep learning methodologies to neuroimaging challenges, in this case, segmentation. These methods have demonstrated great promise in volumetric analysis, and at IXICO we've seen that the use of a UNet architecture with data augmentation can provide robust regional segmentations across a range of scanner protocols for subcortical structures, for instance. This has been presented at several conferences, as shown in the bottom left. We're also currently working on applying and validating these methodologies for specific therapeutics, including the core Alzheimer's regions of interest, such as the hippocampus, whole brain, and lateral ventricles, where we expect and indeed are seeing increased accuracy and sensitivity for efficacy. And we look forward to reporting the outcomes of this work in the future as it develops. With that, I would like to close and hand over to Sonia.
Well, thank you very much for that part of the presentation. I'd like to thank you very much to Richard, Shona, and Roger for that insightful presentation. And now we're going to jump in and do just a couple of questions for the Q&A portion, as our time is almost up. So let's start with the questions here that have already been sent in from our audience members. And the first question looks like it's for you, Roger. The question is, if more substrats for PET become outcome measures, how can we decide on a PET strategy and add trials? AD trials.
Yeah, thanks. Well, I kind of alluded to it a little bit in my talk, but you know, it really depends how the science and the biomarker choices work, I suppose. I guess if we get another new PET ligand, that looks like it'll be affected by a compound, we may drop the amyloid, or we may do, you might do a CSF instead of for amyloid positivity. I think it depends on the science and then the ligands, but it's going to be a tough set of choices.
Okay, thank you very much for that answer, Roger. This question looks like it might be for Richard. Richard, in your presentation, you show, yeah, it looks like it is for you. In your presentation, you show whole brain parcellation as part of parcellation as well as individual regional segmentation. Why do you employ two methods for extracting the volume for the same region?
Thank you. So both methods have their advantages and disadvantages based on context. Whole brain parcellation can provide volumetrics for both regions of interest, but can also be employed in other complementary analysis, such as defining regions in fMRI analyses, as well as DTI analyses, linking up with the volumetrics, providing optimized individual segmentations, as we did for the hippocampus, provides a slight increase at accuracy, but has the downsides that there's no guarantee that neighboring ROIs won't overlap. So you're limited in terms of how you can apply these parcellation schema to other endpoints.
Okay, thank you very much with that as well. Well, it looks like that's it for us. We've come to the end of the Q&A portion of the webinar. So if we couldn't attend to your questions, the team in IXICO may follow up with you after this presentation. If you have any further questions, direct them to the email address that's on your screen, and there it is. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link, and also share this link with your colleagues when they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers, Roger, Richard, and Shona, for that very insightful presentation. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now.